of the future. And I can reassure you, she hasn't been there that whole time because she doesn't look that old. Dr. Bowles has an impressive resume. She earned her Bachelor of Arts in Linguistics and graduated with honors, magna cum laude, from UC San Diego, and has a PhD in marine biology from Scripps Institute of Oceanography in La Jolla. She has for over 4 decades of experience in studying the effects of man-made noise, when we've been making a lot of that, on a wide range of species. In addition to cetaceans and pinnipeds, she has studied manatees, domestic animals, desert tortoises, leatherback sea turtles, kangaroo rats, kit foxes, polar bears, birds, including northern goshawks and spotted owls. Wow. And there's a couple more. We just have a repeat of some other stuff. She did, we lose Ann?
Oh, yeah. Maybe did I go too far? Oh, yeah, leave it gone.
You probably did.
I went too far.
All right, she's back. She's back. Okay, just let's get this introduction out of the way and not take any more chances. She serves on a whole bunch of advisory panels: Acoustic Society of America, U.S. Navy Marine Mammal Commission, NOAA, San Diego Zoo, National Park Service, National Marine Fisheries Service, and the International Union of Conservation of Nature. And yada yada yada. She's done a whole bunch of stuff. She's an awesome. She's an awesome supporter of ACS San Diego. That's not on the bio. So she's an awesome supporter of ACS. I can do it. I can actually do that.
Okay.
All right. And
So thank you very much. So
So what's
you might
It's not showing this on her page.
let me get that. No, that's Zoom. That's Zoom. Go.
Okay, beautiful. All right. So somebody said you can't possibly be that old. Well, I have not been at Hubs for all 60 years, but I've been at Hubs for a scary long time. So there's an awful lot of work that we've done over the last 60 years, and I'm not going to try to reprise all of it, but I am going to give you kind of a personal tour of the stuff that I've found really interesting over the years, and that'll give you a sense of what the organization as a whole has done. So all right, so we started out as the Mission Bay Research Foundation in 1963. So that is the year before the SeaWorld Park opened. So Hubs as an organization was there before the park got to be beyond plans at a bunch of earth moving equipment. And the first things this was chartered by Milton C. Shedd, who was one of the founding fathers of SeaWorld. And he was originally using it for doing research in support of sport fishing, but it grew to the point where it began to publish oceanic sort of pollution abstracts and ocean index. Back in those days, you didn't have the internet to look things up. And so scientists had to compile these abstracts to help people understand what work was being done. And that was one of the services that it performed. So that gives you an idea of how the organization started out. There is on our website a timeline that gives you a whole bunch of details, but this is the quick version of it. What's important is that I moved to San Diego the year that SeaWorld opened. So I've kind of got a long history, and I joined the organization in 1978. So I've seen it from the point where it became a marine park in San Diego, and also the point where Hubs became the Hubs SeaWorld Research Institute in a dedication ceremony. Now, what's important, instead of thinking who are all these people, is to notice the box in the back there. That's the kind of box that you take to the Antarctic to serve as a office space, or to the Arctic or any place in harsh climates. And that was my office when I first came to work at Hubs. That was the bioacoustics laboratory at Hubs SeaWorld at the time of our dedication. And I sat in there, and what did I do inside the box? Well, I'm actually going to step forward one and then I think come back to actually, you know what? Do you have a little teeny light somewhere? Do I have a little teeny light somewhere?
Yeah, here we go.
I don't want to move my phone because I don't want her to get disconnected.
No, we don't want to be, okay. This is a K-allometric sound spectrograph. Anybody who's ever done work with sonograms realize that this is a picture version of what a sound looks like. So you see an example of one of these things over here. This was produced by a colleague. The thing used a little charged wire to burn holes in treated paper to make these pictures.
Wow.
We called it a smellograph because we were causing lung damage every time we spent the morning in there making sonograms. But this is how we did it back in the day, and be glad you don't have to do it now. All right, so the very first year I was there, I began to realize what kind of a place I was in. They were doing a swordfish tagging study on the SeaWorld down around the tip of Baja California, and uncovered 30 killer whales in the process of trying to take and eat a blue whale. This is the first, there's a paper running around now that says that they reported the first time it was seen. No, this incident was the first time that it had ever been seen. Technically, we didn't see the whale killed because the killer whales were sort of eating pieces of it over a 5-hour period, and when it got dark, the SeaWorld had to give it up. But it was full-blown attack, and they had a lot of information about which whales were working what parts of the problem, and it was obviously a very concerted effort on the part of these animals. So that kind of gave me the sense of the coordination and the control that these whales have. This kind of stuff is on a timeline that you'll find on our website that goes through all of the things, and this is just one example of the sort of firsts that I've had the opportunity to see over the whole period that I've been there. So what sets us apart, we're an independent, not-for-profit research organization. We're solutions oriented in the sense of science solutions. We take a non-advocacy approach. We have access to the animals at SeaWorld. But for me, one of the most important things has been a remarkable group of people as well as remarkable animals that we've had the opportunity to work with. So I'm going to toss out some of the characters that I've encountered along the course of my career there, as well as talking about the work that we did. And the focus is not working on health issues or something like that that's internal to the park itself, but to take advantage of having access to the animals to go out and solve problems in nature and to learn how nature works. So another important thing about Hubbs to understand is the legacy of Carl and Laura Hubbs. Carl Hubbs was on the faculty at Scripps Institution of Oceanography. One of his students was Ken Norris, who is basically the father of marine mammal science, probably worldwide, but certainly in this country. And he was an amazingly prolific scientist and has had a whole ton of different kinds of animals named after him. I've got a few examples here. I want to make 2 points. One is you can't see it necessarily off the right-hand side of this picture here on the legacy. I think at home you can see it. There's a picture of Carl Hubbs with his wife, Laura. She was not a homemaker support structure. She was a trained mathematician. She handled his statistics. She was absolutely integral to all the work that he did. And those of us who had a chance to meet her back in the late 1970s are just tremendously privileged to have had a chance to talk with her, because she was a wonderful person and a fantastic scientist. Carl was obviously a pioneer ichthyologist, but he was also a pioneer in something that people hadn't really thought much about for fish at that time, and that was fish conservation. He not only worked on marine fish, but also studied and named fish in these tiny little refugia throughout the Southwest, and was responsible for naming species, in this case, naming a species that went extinct before people figured out how to conserve it. So he was a force of nature himself in trying to get these animals to be properly conserved. The other thing is that he's one of 3 people that I can think of in the whole time that I've been in this field who has had a cetacean named after him. In this case, it is the Hubb's beaked whale. And this is important for a second reason, because I tell every student that comes through, I tell them the story of Carl Hubbs and how the Hubb's beaked whale was named after him, and how it at that time was identified from, a couple of beached animals that nobody really knew anything about. So I had a graduate student who got all excited about the Hubb's beaked whale, and she decided that when she went to work for the National Marine Fisheries Service, that she was going to actually be the person who identified the vocal of the Hubb's beaked whale. Well, the bad news is that somebody beat her out. That paper is just now in press, but in the process, she came across the BWC. This is an animal that is known only from its vocalization. So this is something that happens with beaked whales because they are very deep diving. You don't see them at the surface very much. They're out in the open ocean. So you go to areas and you find these vocalizations. I would play it for you, but it's 35 to 100 kilohertz. You can't hear it, and I probably, it's this little high frequency upsweep that is characteristic in duration and frequency, and it's been picked up, but they had no idea what kind of whale it's coming from. So what she has done is to add several important pieces of information about this through a remarkable effort that shows the whole point to doing bioacoustics. An animal that you can't see, it turns out it only feeds at night, so you're only hearing these little sounds. Basically, sometime in the dusk, but basically it's nocturnal. It's occurring in shallow water, so it's a beaked whale that's foraging in fairly shallow water, which makes it completely bizarre because mostly they're deep diving species. And it's something that everybody's been excited. Let's find out what it is. So she pulled together data from sophisticated buoys that allowed us to do localizations, but she also had data from a guy who rode from California to the Philippines, towing a hydrophone, collecting data the whole way. Equipment from 6 different organizations all pulled together. And what she has discovered, first off, I didn't know where the cross seamount was. So it's in this picture, it's the place that says cross seamount. It's a seamount just off the southern end of the Hawaiian island chain. And this animal was first identified in that location. It turns out, though, that it occurs in these pockets of fairly shallow water across the Pacific Ocean. So now we're looking for a whale with these characteristics. So any of us who does whale watching, it's time to go out there to these very specific locations and try to see if we can find this thing. That in biology circles, there's a prize for that. And the way that they usually recognize them is by actually making a sighting and then getting something like a biopsy dart so that you can get genetic information from it as well. So this was a very exciting process, watching this student go from being just a student to somebody who is working their way through the mystery of a species that most of us would never have an opportunity to know anything about. Okay. Other remarkable things over the course of my life, I was able to participate in laboratory-based studies of a California gray whale named JJ, who was raised from a fountain. She stranded, had been abandoned by her mother and raised to a year of life. We were able to discover that her vocal repertoire was perfectly gray whale-like, so she managed to develop it all on her own. And there were many, many other studies that were done, collected together in this issue of aquatic mammals. And I can't think of anywhere else on the planet I would have had the opportunity to do that. Now this one, my office mate and I stood on top of our desks looking out at the window. We didn't have big windows, we just had a little window at the top so that we could watch This is obviously this is back in the late 70s and early 80s. It's not something anybody would do these days, but when VIPs would come into town, you had the privilege of seeing them playing with the false killer whales. Okay, serious work. My first task at Hubs was actually to study killer whale color patterns. And it was a really important study from my point of view because it taught me, oh, you know, there's an awful lot of genetic diversity in these animals. We think of them as being a species that occurs worldwide, but their color patterns tell us that they're broken down into subpopulations. And in fact, that has proved to be true in the decades since I initially did that study. That was an important aha. The next important aha came from recordings of Antarctic killer whales that my colleague and mentor Frank Aubrey made, and Jeanette Thomas is probably better known in our field because she was the editor of Aquatic Mammals for years and years, and they were responsible for collecting these recordings. And I'm going to go ahead and play one for you if I can get myself to the point where I can see the little clicky thing.
Good job.
Except we don't have audio here.
Yeah, that's not on your, I think so. That's on, that's over on yours.
Okay. Because my speaker is not turned on. That's why. Oh, alrighty. So we'll bring the speaker over here and get it turned on. Let's give that a try. Now,
Perfect.
that warbly little sound doesn't sound anything like killer whales that you'll have heard all over the Pacific Northwest. So regional differences are found worldwide in these guys. At the time that we did this work, what we knew most about what was happening in the Pacific Northwest. But you will hear the rest of the story about that in just a minute here. The other thing that we did, SeaWorld partnered with us and with a geophysical survey company to create a towed array. It was the first time anybody had ever used a towed array for marine mammal survey work. When we were done with the work that we had been funded to do in the Eastern Tropical Pacific and the Antarctic, this array actually went to NOAA, and that was the beginning stages of their now very active bioacoustics marine surveys that went along with visual surveys. And Jeanette Thomas was the one who spearheaded that effort. What else did well? There's some highlights moving forward a little bit backward a little bit in time. One of our very famous starting staff members was Joseph Argel Jr., who was one of the people who discovered that DDT was actually causing eggshell thinning in marine birds. And one of my pleasures over the course of my career has been to see the brown pelicans and the ospreys coming back as a result of the findings of him and other people like him. So I came in at a time when you hardly ever saw flights of brown pelicans coming through Sunny once in a while, but it was very rare to see them. And now in the summertime when they're breeding, they're out there all the time. So that's really exciting. Terry Williams, who is now on the faculty at UCSC, worked to measure the effects of exposure to oil. Our team was the one that discovered, I don't know if you guys can see it, I'm going to wave up here, that Dawn dishwashing liquid is the best thing in the world for getting oil off marine birds and mammals. And that was a result of work that we actually did at Hubs. And they also were part of the response to the Exxon Valdez spill in Alaska. So there's another thing that was kind of a big thing for those of us watching it going on. We had a Center for Marine Veterinary Virology. When Pam started her work, there was one virus known in marine mammals. People were sort of assuming that if it's out there swimming around in the ocean, it must be fine. It doesn't get sick, it's fine. And by the end of her stint in this program, they had identified something like 14. It's up to more than 30 of them at this point. So yes, marine mammals get viruses, and they have detected coronaviruses in these guys, which is obviously relevant to us nowadays. So that was another really important program. Okay. The other thing is that our ocean health program has hosted an awful lot of workforce training in what we call One Health, which means that we're interested in marine life because of their health, but we're also interested in the fact that there is feedback between human health, health of wildlife, health of people's pets, health of wildlife. It's all interconnected. And we've had a long series of really great students under these programs who have been learning about the value of understanding wildlife and trying to maintain health for all of us worldwide. We also had a couple of very good biologists working on the pinnipeds of the California Bight. When in 1911, the elephant seal practically went extinct. There were 6 young born in the year that they found a colony of them in 1911. By 1982, when our investigators did the first series of counts out in the California Channel Islands, there were 25,000 northern elephant seal pups. They're now up to about 42,000, and the population as a whole is up to 187,000. So we have basically stood witness and collected data on the recovery of pinniped populations throughout the California Bight over the course of our existence as a research organization. Are we just in San Diego? No, there is also a Florida research laboratory for hubs. They're a fantastic team of people. They're located on the Indian River Lagoon in land that was donated to us. So they're right there in an area that has what we now know to be an endemic population, well, actually endemic subpopulations of bottlenose dolphins running from up near where Cape Canaveral, I guess it's Kennedy Space Center is, and all the way down to the entrance to the Indian River Lagoon in the south. And when we first started doing work roughly in the 1970s, nobody really knew that it was a resident population. They didn't know much about its biology. They didn't really know how many there were. The idea originally was, okay, we'll freeze brand them, and like we do with terrestrial animals, mark them, and we'll be able to follow them for years. It turned out not to be such a great experiment, but there were a few individuals like this number 56 that was spotted 35 years later running around with this. But obviously, we're not using that approach at this point. They have shifted over to photo ID as a way of identifying individuals. In the 80s and 90s, aerial surveys, but they weren't systematic, and you have to do this work systematically. So the estimates that they came up with are really variable, somewhere between 30 and 1,061, but that's not a very helpful number.
Tight though, tight.
So now our team, in 2001, there was an unusual mortality event in the Indian River Lagoon, 61 dolphins stranded. They knew something was wrong, and they realized we don't even have a viable abundance estimate for this species. We don't even know how bad that is. So that's when our team started doing systematic aerial surveys for bottlenose dolphins, and that work continues to this day. I'm going to describe a little bit about how the techniques have become more and more sophisticated over time, but even with just decent aerial surveys, they were able to show that the population shifts during different seasons, that they are using the southern end of the lagoon a bit more in the wintertime. And the big question by the end of this period, which was, I don't know, roughly 2011, is to what extent are they going out into the outside world? So what they found is, now that we have a really tight estimate that runs between 980 and 1,100 dolphins, they're resident in the lagoon. They're occupying everything from the Banana River south to the end of the Indian River Lagoon. And that means that whatever it is that we dump into the Indian River Lagoon, that's what they're getting. So we've been working recently to collect morphometric data using drones, where participants, we basically run the NOAA stranding network for Brevard County, which sees one of the highest numbers of live stranded cetaceans anywhere in the country because the water is very shallow. And they're doing this photo ID work. They're also now starting to find evidence that as the Indian River Lagoon seagrasses have died back, they've got a big pollution problem, and it's the quality of the environment is worse and worse. They're finding that they can actually see this in the body weight of these bottlenose dolphins using morphometric data collected from drones. And they're also now starting to collect breath samples using drones so that they can get DNA and health information from them. So it's gone from being a once in a while we do a survey and don't get very much information to something that's very sophisticated now. Yes, we are using snot sampling. It works beautiful.
Snot bots.
Snot bots. Yeah, and when the animal blows and it's got a
Yes.
Yeah.
Well, they're doing it for the killer whales.
So we've started doing this on now. In the interest of time, because we had all this delay in the beginning, I'm not going to try to wax eloquent about my favorite topic, which is vocal learning in killer whales. Now, if you want to get the whole lecture, you can stay after, and I will happily run through this story. But basically, we have provided the strongest proof that you can have with a social mammal that killer whales learn vocalizations, that they actually have quite good capacity to imitate. We had one that was socializing with bottlenose dolphins that not only learned sounds the dolphin had learned from human beings, but also learned to imitate the dolphin's signature whistle reliably enough that you could tell what it was. So they have the capacity to do this, and people will ask, well, so if they're that good, then why aren't they doing what bottlenose dolphins do, which is to imitate bridging whistles and all this kind of stuff? They do it, they do it to raise hell, basically. This is because for killer whales, this is a social thing. You are bonded to the individuals that you learn to imitate calls. It is about the quality of the relationship, and that is the relationship that precedes the learning process. So it's a very good way of assessing the interactions between these whales, which was an outcome that we hadn't predicted in advance, but it's turned out to be very useful looking down the line at assessing social interactions of other populations as well. So does that mean that killer whales out there in the world are changing their vocalizations all the time? We already went through that one. Well, for that, I want to step back for a minute. Another thing that I did fairly early on in my career was participate in a study that Steve Leatherwood, who was kind of the godfather of marine mammal cetacean field research, really. People were doing this work in the Pacific Northwest, but they didn't really have an idea of what was happening with killer whales anywhere else in the world. So they started a systematic effort to assess the populations of killer whales in the North Atlantic. And one of the things that he did, based on having all these bioacousticians running around, Steve, could we get some recordings of these guys? Was actually to get the field biologists to go out and collect recordings. And he had two, Thomas Learholm in Iceland and Johan Sigurjonsson in Norway, were successful in doing this. By comparison with what we can do nowadays with sophisticated remote recorders that'll run for months, it was pretty pathetic. It was, 2 45-minute cassette tapes with these recordings on them, but they were beautiful recordings of large groups of whales feeding on herring. And so we were able to analyze that stuff and get stereotyped calls that are completely typical of killer whales everywhere we've had a look. So one of the big questions is the kind of stability that we see in the Pacific Northwest in the relationships of the whales and also in their vocal repertoires. Is that going to show up somewhere else? Well, there's no other place in the world besides this Icelandic research program that has actually been asking that question in a long-term kind of way. So without really meaning to, we participated in the earliest recordings for that program. And just this year, so I'm kind of bookcasing my career. My first bioacoustics papers ever were written as part of the North Atlantic killer whale program. And my most recent bioacoustics paper has been written on killer whales from the same Icelandic population. And it turns out that 4 of the calls that we identified back then are still in use around Iceland, and that the population of herring apparently made a big shift between the earliest recordings and these most recent ones. But since the move, the whales have been producing very stable repertoires over the whole of Iceland. So decades, over a period of a couple of decades, which means it seems to be a killer whale thing. Of killer whales? Probably not lots of generations. The adults can live 50, 60, 70, the females especially can live to be a bit older. But you have calves being born in a healthy population. You have calves being born every, say, 3 years, something like that. So they're coming up, they're learning the repertoire, and they're somehow keeping it very stable over this time. So this leaves us with, now the question to me is, okay, if they're generally really stable, but we know they can learn, then when do the innovations arise? Under what conditions do the innovations arise? And that's the question that I would like to look at for the next 60 years type of thing. Okay, and just so that you can be convinced that they're actually doing this, I am going to play you some killer whale. So this is one of the ones that is, we identified that is still in use. These. All right. I'm going to cheat. Is it going to play?
The speaker on?
Okay, that's call type. .4, that's the one. So that's how killer whales sound. And when people say they're screaming, yeah, they sound like screams because they're really high frequency. But these are their regular, the regular calls in their dialect. All right, just for fun, one other call type. It's going.
Short.
Yep. Okay, so that gives, and that also helps you to appreciate why it is that I said earlier that this is a really, really strange call by comparison with what we expect. These.
One more.
I think that's a really strange call.
Yeah, it was a good call.
That's about the most exciting. You know, I think the PC Ferry has really decided to go after
that
me
one,
tonight.
somebody was unmuted. That was very good. Very well timed, though.
Hope you feel better. Oh, now we're dull. What's going on?
Well, now we're gonna get to be smart and just do
we
this
lost.
over again.
We lost you here.
Yeah.
Screen sharing is stopped. Okay,
so if
but
you
we're
can-
still okay.
We're still okay. and I will get it opened up again. I promise. All right. Actually, I'm going to go back to where we
You
belong.
don't see it?
Okay.
While we're waiting for that,
I might just mention that we are recording this, and we do have a YouTube channel. So ACS San Diego has a YouTube channel, and it takes me a couple days to process it, but I will
Okay.
put it up on our YouTube channel.
Okay, and I'm just going to move along because what I'm hearing coming out of here is people and not killer whales. All right, so where do I want to go in the
Okay, wait.
future?
We're not seeing your
Oh,
presentation.
because I haven't shared this yet. That's because I need to share it. Good morning.
She should down at the bottom, share screen.
Out of the bottom, share screen. There we
Down at the bottom of your Zoom,
Yep,
share screen.
it says share, right like that.
There we go.
Is that
Okay,
better?
back.
Yep. And then play.
Okay, there we are.
Perfect.
Oh,
All right,
awesome.
yeah,
I'm sorry.
yes.
Are you able to match the vocalizations to particular behaviors? Or you can talk about that later?
Well, that one is a long and complicated story. I can tell you that in this field, you go forward little teensy weensy steps. Established that the bubble trails that you see in these guys when they're socially wound up are accompanied
Eating.
by a particular type of call.
Signature whistles.
That's a theory. That's the theory that we have. They are the kind of calls that have two simultaneously moving sound sources in them. So killer whales have two independently functioning sound sources in a number of their calls, and they tend to bubble the ones that have that second sound source. And they do it. This is the thing that blows me away. They succeed in producing this call, running air through the same passage that they're using to generate the sound. So this big flow of bubbles going out of their blowhole, and the acoustic characteristics of that sound do not change detectably. We have sat down and measured calls that are bubbled and calls that aren't bubbled, and you can't tell the difference.
Is it like a ventriloquist not moving their lips?
Maybe.
We don't know on the inside how they're doing it. All we know is that that passage to the blowhole goes right past their monkey lips, what the equivalent of our vocal folds.
Mm-hmm.
So that's an exciting thing to figure out in the future. All right, and I have moved outward in the marine environment a little bit for a number of reasons. This little fish is a juvenile white sea bass. I made a big embarrassing mess of myself by sending this to a colleague. What the heck is this? Because it doesn't look like any of the pictures of white sea bass I've ever seen. He said, oh no, it's lit up. So this very bright light color with the very dark bands is typical of these guys when they're all excited from having had something to eat. So this is a white sea bass swimming around in the SeaWorld marina who just thinks that the food's great. So we were just testing equipment there. We're in the process of trying to learn something about how animals interact with stuff that people put in the ocean. My interest obviously is lines because we've seen so much entanglement in the work that we've done both here and in the Florida hubs. So we got a great picture of this little guy, but I'm also going to play you a video because one of the things that we're interested in is how animals interact with these lines. The answer is that we don't particularly know, but this is a GIF. You can see the bat ray. The bat ray contacts the line, and he hangs a U-turn going in the other direction at high speed. You do that, and sometimes you get yourself wrapped up. Or if you're not, like a bat ray, which is probably shaped to have the line slide off and pretty well, but if you're something else, you get yourself entangled. And we got that in 14 hours of observation. Now, that's a sample of one, so I'm not going to try to extrapolate to an encounter rate, but it does leave me with the sense that if I were to do the same exact exercise, this is a hyperhemispherical camera, so it sees basically more than 180 degrees above itself. If I were to put that on lines, I might be very surprised at who comes into the area and who interacts with them. And that's what we would like to do at this point, is get some real numbers for that. Okay, so that was the personal view. Just to make a point, we have worked all over the SeaWorld Park on a whole range of species because we've had access to them. Sometimes it was like this that I just described. So me doing tests in the SeaWorld Marina, tests on tracking equipment, tests to see how fast these guys can swim if we condition them to swim as fast as they possibly can. All of this kind of stuff is possible when you have access to the animals. And so we've taken advantage of that over the whole of our history to try to get information about a wealth of issues that these guys face out in the wild. If you're interested in the research projects that we've done in the greater the world, so we also do a lot of work outside, that's our job, is to take advantage of having access to them to learn stuff for the outside world. So that's what we've done. I've got a circle around my most remote site, which was Heard Island in the southern Indian Ocean. I was a part of the Heard Island feasibility test, and we have a picture of Walter Mott standing next to one of his sources in this as a part of this study. So that's our organization works with SeaWorld, but we also do field work all over the planet. And we've had a lot of really, really amazing partners over the years. So this is just a subset of them. And you guys have heard, I'm not telling you about our fish hatchery or the sustainable seafood program, because you guys have heard a bit more about that in the last few years. That's why I tried to keep it more kind of my own story. But we're certainly continuing to be interested in that. And Danielle Halsey, who is our Chief Science Officer, has also given a talk recently on the work that she's bringing into us on billfish.
And then we did the field trip too.
And you've done the field trip. So you've got a pretty big overview of the organization now. And aside from apologizing for all of the technical difficulties, I appreciate your attention.
So if people want to online want to ask questions, put it in the chat and I'll read it to Ann. The people here on the boat, we'll kind of take turns to see what's going to happen. And Ann, if I could get you to repeat particularly the questions here on the boat.
Okay.
Because I think I'm projecting loud enough to be picked up on your computer, but some of these folks have quite little voices. So anybody have questions? Questions?
Yes, ma'am.
And I love the research you guys did on entanglement and how the manatees were hugging the line when you thought that we were. Can you talk about that a little bit? Because that really hit my heart.
She's asking me about a study that we did studying the interactions between marine mammals, and it was simulated fishing gear because we were doing it on SeaWorld's animals, and obviously we aren't going to hurt their animals in the course of doing our work. But there were a couple of species that gave us some real ahas. She raised the manatee. Florida, the Fish and Wildlife Service in Jacksonville, Florida, asked us, based on experiments that we had done on 6 species of cetaceans and pinnipeds at SeaWorld, whether we thought that these Duquesne netmark pingers, it's a little thing that produces a 10 kilohertz pip, and for reasons that we're still trying to figure out, a lot of marine mammals think they're obnoxious, and so they won't go near them. Well, manatees don't have very good vision. They have a sense of touch, and they have hearing. So when they hear something weird, they want to go and touch it. Exactly the same way you guys, if you hear something weird, what the heck is it? You try to look at it. And so I have a great picture of a manatee responding to tests with this pinger by actually swimming over to the thing, grabbing it, and putting it up against the array of sensory hairs that he has on his body.
That did help.
We could probably go back with the lights on.
Okay, yes,
Yeah.
why don't we do that?
If you don't mind, thank
No,
you.
no, that sounds great. So you can have that
Oh,
back.
it's-
So that's fine.
Yeah, so that was really trying to figure out what it is.
So in other words, the manatee, in order to figure out what something was, they had to touch it, which of course is exactly the opposite of what we wanted to have happen. So for those guys, the way to solve the problem is to actually make the line stiff enough that they can't get themselves wrapped up in it. And we have 2 line types that we've identified now. I've just written a proposal asking, can we have feasibility funds to go out and test this stuff actually on manatees in Florida? It's not a huge problem for them, but at the same time, it's a lot of effort to go out and rescue them. The Save the Manatee Club does this every year, puts hundreds of hours into rescuing them. And it's a source of misery for the animal. A lot of them, hundreds of them, have scars on them from interactions with gear.
Yeah.
So if we could damp it down with a simple change in the gear, that would be the best possible way, and that's what we would like to do. The other thing that we learned from that is that some species of marine mammals are quite intolerant to little high frequency tones. It turns out that little porpoises don't want to have anything to do with high frequency tones. The pinnipeds would get used to it, but the porpoises, we did a series of experiments to see if they'd habituate. The first couple of days they ran around and rooster-tailed and did all this kind of stuff, saying that it was surprising for them and they didn't want to listen to it. By the 3rd day, they'd see us coming in to set up our equipment, and they were in the refuge area that we always gave them during these, they always had the option of not having to listen to it, and they're in the refuge area. And they're not coming out until you get that thing out of our pool.
So we have a couple of questions from the online group, and then we'll come back here onto the book. Laura is asking, is it hard to remain neutral on issues such as captive killer whales?
My job, well, first, my job is to learn what's going on and to tell whoever's asked me the question as accurately as possible what the answer is. So I try really hard not to get sucked into these political debates. It's not our job as an organization. Our job is non-advocacy. Our job is to look at what's actually going on. And what I've found is that a lot of times the debate is taking place in some imaginary land that doesn't have much to do with what's actually going on at the level of the animal.
McKenna wants to know what led you to decide on researching marine mammals, specifically whale species.
Well, I was one of those crazy kids that wanted to, I read everything John Lilly ever wrote. This was the generation, and I went to Hubs going, I want to learn to talk to the dolphins. And my advisor said, I'll tell you what, why don't you study emperor penguins for your thesis? which I did because it was really good advice. But in the course of doing this, I started understanding more about these killer whales, and it turns out that they are the closest model to the way that human, not language in the sense of talking about physics or anything like that, but in the sense of how they develop it, they are the best model of any species that I know of. So for me, it was an absolutely perfect way to go back to what I had wanted to do when I first got into this field, to do this research on vocal development in the killer whales.
Any questions from here? Leanne, our infirmed president, is asking, what is the current population of the Hubs beaked whale? There you go.
Well, my hope is that Jennifer will have the opportunity to find that out. She is doing a fantastic job with NOAA Fisheries. The answer is that we know so little about these guys that I couldn't even begin to answer that question.
Yeah. And one of our board members, who also has got child issues and stayed home today, our lovely George and Marissa, can you discuss the difficulties and expense of deploying hydrophones and analyzing the results? They seem like they're such a great tool for research, but it seems like such a small number are deployed. It must not be easy. It must not be as easy as it seems. Do you think that technological advances will likely result in more being deployed in the future?
Yes, sir. It's already happening. I've been watching the trajectory of this, especially in the last 10 years or so. We now have instruments that I would have given anything to have when I first got into this field. Little tiny thing called a sound trap that you set up, you put it in the water, it runs for 10, 15 days, or slightly bigger ones, you set them up, they run. Right now, the big challenge is not the instrumentation for collecting the data, it's the processing is the huge challenge.
Yeah.
And that's getting better. I think, to me, for AI, forget advertising, forget cheating people, forget political, all of that stuff. AI is for biologists to make it possible for us to process the audio and video that we need.
It'd be amazing tool.
And drums too.
Yeah.
And it's already, we're at the cusp of that actually being extremely useful now.
Questions here on the vote? Leanne's, okay, go ahead.
I thought I just read something about whales and they call themselves, they have names.
Dolphins have names.
So they actually call their name. Hey Joe.
We don't know all of the message that's involved in it, but they certainly call each other. They can imitate one another's, what they're called signature whistles. It's not really a signature because other individuals can use it, so it functions more like a name. They use it when they're trying to figure out where everybody is. So call and they get an answer, call and they get an answer, so that you know who you're going back and forth with. They have like a special baby voice that they use with calves when they're working through the process of learning this stuff. Yeah, it's an amazing system. We don't know how much that generalizes to other species because the bottlenose dolphin is really the best understood in this area. She's just made the suggestion that maybe some of this is going on in the killer whales. I think there's a level that that's true. We just don't have the data for it at this point.
Another one from the Zoom audience, again, Leanne, she's on fire tonight. Were you able to follow JJ after her release?
We were able to follow, we weren't able to follow JJ for very long because she scraped off her transmitter really, really quickly. Part of it was that
Did she
transmitter.
also have a freeze patch? Was she the one
She
that
did.
freeze patch, or was
She
that
did have.
Gigi?
No,
She
that
did
was
have.
Gigi.
That was Gigi. She had markings on her that would have enabled us to recognize her for a while. If somebody ran into her today, I really don't know that they'd be able to do that.
Very cool. Other questions here on the boat? And thank you, thank you. It's always a pleasure. You've come many times and it's always really interesting. And thank you. I'm sorry we had all the technical problems, but it turned out to be an absolute delight. So
Well,
thank you very,
you
very
guys
much.
are a wonderful audience, and I always enjoy talking to you.
Thank you very, very much.
You bet.
We will shut down here. Okay, now everything's off the record.
You got